So today's lesson in our unit Cells and Systems is topic 2 on microscopes and cells. All right, so early microscopes. So we have Anton van Leeuwenhoek. He was a Dutch linen merchant from Holland, and his hobby was making magnifying lenses. So he started making these instruments called microscopes, which were able to magnify objects up to 300 times. So whenever we see the 300x, that is referring to 300 times. So he was studying blood, he'd look at pond water, and even matter scraped from people's teeth or his own teeth. And so he became the first person to observe organisms made of only one cell. And he called these single-celled organisms, he called them animalcules. All right, so then we have Robert Hooke. He was an English scientist, and he was making microscopes around the same time as Leeuwenhoek. And he used a microscope to look at a thin piece of cork from the bark of an oak tree. And he saw these tiny box-like compartments. And he described those little boxes as cellulae, which basically means little rooms in Latin. And it was his descriptions that led to our present-day word, the cell. All right, so cells in all living things. So eventually we had other scientists using microscopes as well, and they were studying microorganisms and parts of plants and animals. So Matthias Schleiden, he was a botanist, and Theodor Schwann was a zoologist. And in 1839, they combined their observations, and they made their hypothesis that all organisms are composed of cells. And they suggested that a cell is the basic unit of life because all functions that are carried out by living things are also carried out by their individual cells as well. Then we had Rudolf Virchow, and he contributed his ideas to those of Schleiden and Schwann as well. And they formed the basis for the cell theory. So the cell theory states that all living things are composed of one or more cells, and cells are the basic unit of structure and function in all organisms. Okay, microscopes today. We have the compound light microscope, which consists of two lenses, which give a greater power of magnification. So we can magnify objects today as much as 2,000 times. There's also electron microscopes. These use beams of electrons instead of light. So basically electrons are bounced off the sample, then enlarged to form an image on a screen. The first one was built in Germany in 1932, and that one was able to magnify 4,000 times. A valuable tool, in 1938, the first practical electron microscope was developed by two Canadians at the University of Toronto. So James Hillier of Brampton, Ontario, and Albert Prebys of Edmonton, Alberta. Their electron microscope was able to magnify 7,000 times. And today's electron microscopes that we use today are able to magnify 2 million times. All right, so we're just going to watch this quick video. All right, so next we are looking at the parts of a compound light microscope. So we'll just start here at the top left. So first off, here we have this part. It's called the rotating head. It contains mirrors, and it allows the body tube to rotate 360 degrees. This here is the body tube, passes light from the head to the eyepiece. So this here is the eyepiece. Note here, the eyepiece is also called the ocular lens. This is what you're looking through. This is where one views the enlarged object. It also contains a lens inside. It's called the ocular lens. and it further magnifies the specimen by 10 times. This here is called the arm. That's part of where you would hold the microscope. It also supports the lenses, mirrors, and body tube. So one hand is always holding the arm, and then your other hand would be holding the base down here. That's how you would carry the microscope. All right, so next we have this coarse focus knob, the larger knob here, that moves the stage up and down. It's used to find the specimen under the low power objective lens. Then you have the fine focus knob. So once you find your specimen using the coarse focus knob, then you're going to use the fine focus knob. All right, and that'll make small focus adjustments, especially when you start using the higher powered objective lenses, which are here. I'll talk about those in a second. The light source is here. It provides the light. It'll shine light through the specimen. The power switch to turn it on and off. This here is the base. So that's the second part that you hold when you carry a microscope. It contains the electronics. It supports the microscope. So like I said before, one hand is under the base, the other one is holding the arm. This here is the diaphragm. By rotating that, you can control how much light is passing through the stage. This is the stage here. That's where you place your slide. It also contains the opening that allows light to pass through it. And then these are these objective lenses that I was talking about. So here, this microscope, I believe, has 3 of them. So the high-powered lens is the blue stripe. It magnifies 40 times. The lower power lens, the yellow stripe, magnifies 10 times. All right, and keep in mind that the eyepiece is also magnifying 10 times as well. All right, so there's a couple of videos here on how to use a microscope. So I'll just pause here and put those on.